Welcome to this lecture on transplantation and how the immune responses are involved in graft rejection or graft acceptance reactions. To understand this lecture, we will go through some basic studies that were done earlier and were also covered in the classes on major histocompatibility complex and how inbred strains were made. Later on, we will go briefly into some of the ways that these rejection reactions can be controlled in order to facilitate the acceptance of skin graft. After all, as we realize now, the availability of different grafts, whether organ or skin grafts, depends upon the availability of suitable donors, and donors are very hard to come by. Even if they are present at a particular time and location, the matching of the tissue is very important in order to facilitate acceptance of skin grafts. Therefore, there seems to be a tendency for taking grafts from cadavers rather than from living donors, although living donors are preferred. So, some coverage of some of the basic aspects of why tissues or organs are rejected will help us understand some of the strategies that are being employed for enabling the graft acceptance, so that more grafts can be organs can be transplanted to those who require it. So, basically to recap, some of the studies that were done in mice earlier was the fact that different inbred strains of mice, individuals belonging to different inbred strains of mice, actually reject skin grafts if they are grafted on to the back of the mouse. Usually they use skin on the back or a piece of tail that has been got from the tail and then graft it on to the tail of the other inbred strains of mouse. All of the studies or the lectures that were given about inbred strains of mice must be reviewed in order to understand this slide. And basically here what I show is a black mouse. An example of a black inbred strain of mouse is C57 black 6, and then you have a red colored mouse here, let us call it as the white mouse or the albino mouse, which is characterized by BALB/C or NIH mice as inbred strains of mice. So, if a piece of black skin were to be grafted on to the white mouse, here it is called as a strain B recipient. A recipient is one that receives the graft, and a donor is one that donates the graft, whether it is a skin or whether it is an organ. So, when the strain A mouse donates this graft and this is sutured onto the back of the white mouse, normally this black piece of skin, actually the word that they use is takes. The word that is used is takes. Just one moment. So, they say the graft takes, which means that the graft gets fused onto the other piece of skin, the circulation is restored, and therefore, the cells inside the graft actually start to proliferate and then actually fuse onto the background skin. Now, after a period of 10 to 14 days, something ensues, which is mainly a T cell mediated response, where the T cells are come to recognize that this piece of skin is expressing foreign antigens, foreign with respect to the major histocompatibility complex. That is because the recipient cells or the antigen presenting cells have actually taken up some of these antigens that are there in the piece of skin that was grafted, and they have been presented via dendritic cells to the T cells of the recipient. In addition to that, there are other aspects that are recognized by T cells, which we will go into later on in the coming few slides, and that has to deal with the recognition of the major histocompatibility complex class 1 MHC. Once this graft is fuses onto the background skin, the T cells that are now activated and are actively proliferating and have recognized the presence of this foreign skin start to react against the skin and activate inflammatory processes, which cuts off the circulation to this piece of skin. Inflammatory processes are the same kind, generally the same kind of processes that occur when you have inflammation going on during infection or other kinds of immune responses. So, within a period of 10 to 14 days, this graft, because the circulation has been cut off, the graft loses its circulation and becomes dry and slowly starts to die and necrose, and after a period of time it becomes dry and the whole piece of skin just falls off from the back of the grafted mouse, leaving an empty area and exposing the muscle underneath. So, therefore, this period of primary graft rejection takes about 10 to 14 days in the mice, which is due to a primary anti-graft response. Now, if one takes lymphoid cells, especially T cells, from this mouse and adoptively transfers it, in other words, take these cells and inject it via the tail vein into a naive mouse or a mouse that has not been exposed to this black skin, after which you take the same piece of skin from the black mouse and graft it onto the back of this white mouse, you find that instead of 10 to 14 days, the graft is rejected within 4 to 6 days, reminiscent of our lectures, signifying that this is a memory response. And this memory was actually transferred because these lymphoid cells, especially studies have been done to show that a majority of these reactions are due to the transfer of T cells. Although antibodies that are raised against this piece of skin can also mediate graft rejection reactions by accelerating complement mediated lysis as well as other kinds of reactions. So, this acceleration of reaction is due to a secondary response. So, this is the basic graft rejection response that is studied in mice, and these are the studies that were used in order to characterize the H2 complex or the MHC complex in the mouse. Now, all this led to the concept, of course, that the self is recognized within the body's immune system, and therefore, these cells, when they recognize something coming from outside, is considered as foreign. So, the concept of foreign, self and non-self, now needs to be defined in a different way, and that is something to do with the MHC per se as well as the pathogen coming in, and we all know now that the pathogen that infects an animal can be presented in two different ways, and that is the class 1 mediated response and the class 2 mediated antigen presentation. So, a class 1 mediated antigen presentation involves endogenous virus or pathogen replication within a cell. So, if a virus invades or infects a cell and the virus proteins are made within the cell, these cells are processed and presented by the antigen presentation machinery via the class 1 molecules. Just to summarize it, the viral antigens are broken up into smaller peptides, and these peptides are transported on to the MHC groove of the class 1 MHC, which is expressed on the cell surface, and these peptides are usually 9 amino acids in length, as opposed to viral toxins or viral antigens that are circulating in the serum are taken up by antigen presenting cells by micropinocytosis or endocytosis, or during phagocytosis of the pathogens, so that they are treated as exogenous antigens, and the processing and presentation occurs through a different pathway called as the class 2 antigen presentation pathway, in which these different antigens are again clipped into smaller fragments, which are then loaded on to the class 2 type of MHC molecules. In other words, the class 2 type of antigen presentation involves exogenous antigen presentation, because the antigen is derived from outside, and whether it is class 1 or class 2 mediated antigen presentation, the ensuing immunological events are different in terms of activation of different kinds of T helper cells and the kind of pro or anti-inflammatory responses or cytokine release that happen later on due to this stimulation. So, just to recap some of the lectures that were done earlier, or to recap some of the information that is required to understand this lecture, is that the adaptive immune response or the acquired immune response requires TB cooperation, and this TB cooperation requires this antigen presentation, and antigen presentation of course requires the participation of MHC molecules, which are your self signature molecules and distinguish self from non-self. So, these presented antigens then tell the B cells to make, or activated B cells to make antibodies against different kinds of antigens. So, how does a virus or a virus infected cell get recognized by this sort of acquired immune response? So, when there is a transplantation process going on, or grafting in grafting procedures, one has to take into account these sort of responses that the T cells orchestrate to the foreign MHC or the foreign incoming organ. And of course, because this sort of acquired immune response is involving a variety of cytokine release as well as chemotaxis of various kinds of cells, one has to look at how these kinds of processes can be inhibited so that the graft survives. In fact, the later studies or modern day studies have revealed that immunosuppression is a better way to go about trying to make the graft survive, rather than to try and find 100% matching organ for the recipient, because 100% matching is only possible in identical twins, and this is not possible with any other donor, although siblings and relatives can be better donors than other kinds of donors who are not relatives. So, this is the concept of tissue typing. We will come to that in a little while. So, if you look at how T cells and B cells cooperate, they cooperate by the secretion of cytokines. So, therefore, if you alter the secretion of these cytokines, then try to use agents or chemicals that will suppress the formation of cytokines that activate T cells and B cells, then you have a muted response to the incoming organ. So, that is one way of trying to combat organ specific allotransplantation reactions within the recipient. The other way of course is the discovery of regulatory T cells or Treg cells. Can these Treg cells somehow be stimulated in order to suppress the ongoing T cell B cell interactions as well as T helper responses. So, these are the two ways by which these regulatory mechanisms can be tinkered around with in order to enhance the period of survival of an incoming graft when it is not a 100% match. So, some of the definitions are in order, and as we saw earlier, in a mouse strain whose individual members possess homozygous chromosomes, that means all the chromosomes are identical with the exception of the Y chromosome. Now, these inbred strains produce identical homozygous progeny when they are mated with each other, and these inbred strains of mice can be obtained by brother sister mating, upon which is based upon a statistical principle of mating more than 10 to 15 times brother sister matings. As opposed to an inbred strain of mice whose chromosomes are identical except for the Y chromosome, wild mouse populations as well as human populations are not inbred, and therefore, there is a wide variety or a wide variation in how in different kinds of the sequences that are present within the genome of individuals, and therefore, they cannot be considered as inbred. So, in order to understand this graft, because we are going to deal with grafts, we need to understand what the terms that are used with regard to grafts. And here we see what is a syngeneic graft. A syngeneic graft is the transplant of tissue from one member of a species to another genetically identical member of the same species. An isograft is a transplant between different parts within the same individual or member. In other words, if I take a piece of skin from my left hand and put it onto my right hand, then it is an isograft. This is one of the procedures that is followed in burn victims. So, when patients come in with burns, serious burns, they take the piece of skin which from the unburnt area of patients and then try to grow it within the lab on synthetic media in order to increase their mass. If that is not possible, they take that piece of skin and put it onto the area where there are burns. This is what plastic surgeons do in order to see how beautify the area which is burnt. In other instances, during bypass, where you take the veins that are from the leg and then put it on to the place where it is required in the heart in order to take out those portions which are blocked and therefore restore the coronary circulation, so that the heart can keep pumping. The other term that needs to be understood over here is what is called as an allograft. What is an allograft? Allograft is a transplant of tissue from one member of a species to another genetically distinct member of the same species, and this is where organ transplantation in humans comes in. For example, if one takes heart from my body or a piece of kidney organ from my body and put it on to another person, then you have an allograft. So, these allografts are usually expressing our self signature molecules. In fact, the class 1 MHC antigen is ubiquitously expressed on all cells of the body, except some of them, some of the cells like the reproductive organs and the brain. So, genetically distinct members of the same species are allogenic to each other. So, we in the human population are actually allogenic to each other, and therefore, grafts that are taken from individuals are allogenic. Now, the other one that needs to be defined is this xenograft. The xenograft is the transplant of tissue from one member of a species to another genetically distinct member of a different species. For example, mythologically we always celebrate Lord Ganesh festival, and there you have actually a xenograft, the head of an elephant on top of a godly human, let us say. So, these are actually genetically distinct members of different species. They are said to be xenogenic to each other. The reason why this term needed to be defined is that nowadays the medical fraternity is resorting to more and more of trying to see if you can take organs from pigs and graft it onto humans, because pigs in many respects are supposed to be closer in respect of molecular expression, surface expression of different kinds of molecules is supposed to be closer to humans, in which case immunosuppression becomes a very important part of transplantation. Also, they are resorting to pigs again because trying to get a tissue match and trying to get a donor organ all the time when you require it is always not possible. So, the idea is to try something that is xenogenic, like from the pig, and enhance the life of that individual for how many ever years that the medical people can. So, going on, some other points that we need to consider is that the MHC is polymorphic. The presence of strain specific antisera has been these are all the reagents that were used to show the presence of cell surface antigen, and they showed that this cell surface antigen is actually highly polymorphic. In other words, there are alternative forms of the gene or alleles, and this is the concept of the self signature molecule of the MHC complex. And if one were to compare the primary amino acid sequence of the MHC molecules that are present within two different individuals or human individuals, you would find that this primary amino acid sequence as well as the primary nucleotide or the nucleotide sequence of that gene is going to be very different, and that is what makes the MHC polymorphic and it being a self signature molecule. So, the important concept arises now in order to understand the concept of anti-graft rejection reactions, and that is the expression of these molecules on the cell surface is codominant. Codominant meaning that we need to remember that we inherit half our traits from our parents, half from the mother or paternal maternal and half from the father or paternal. So, you see even in the MHC, the MHC molecules, half of them are derived from your father and half from the mother, and both of these molecules, the MHC alleles that are derived from the father and the mother, is actually known to be expressed right from the very early embryonic stages, somewhat later than 8 cell stages. However, the fetus is never rejected or not subject to harmful immune reactions by a variety of mechanisms, which is a field in itself. So, going back to codominancy, which is meaning of course, that since you derive your MHC antigens from your father and mother, your antigens are both the types of antigens that are coming from the father and the mother are expressed codominantly. In other words, both of these molecules are expressed on every cell surface. So, if you have different class 1 alleles, then these different alleles are expressed on your cell on every, since they are ubiquitous, they are expressed on most cells of your body. So, both maternal and paternal gene derived proteins are expressed on the same cell. This is the concept of codominant expression. In outbred populations, the progeny are heterozygous, whereas in inbred strains of mice, the maternal and paternal H2 loci are homozygous, because their genetic sequences are identical. Therefore, the MHC molecules that they express will have identical structure as opposed to a outbred population or a population that is not inbred. In fact, this is how the MHC function was discovered, because inbred strains of mice were bred. So, all primary, secondary and tertiary structures could be slightly different in different individuals. Primary of course, definitely being different, but there is an overall what you call as an MHC ness to the MHC molecule, and that is where you have what you call as an allorecognition phenomena, which we will come into in the next few slides. So, going to the MHC genetic map, you find of course, this genetic map shows you that there are different kinds of MHC antigens, as I told you, class 1 and class 2 antigens. Class 1 being signified by the red rectangles over here, called as the A, B and C. So, HLA-A, HLA-B and HLA-C are class 1 molecules, as opposed to class 2 molecules, which are shown over here. So, you have HLA-DP, DQ and DR. So, you have DQ over here and you have DR and DP. So, you have different alleles. So, there are since they are polymorphic, they are going to have different sequences, and therefore, different alleles are going to be expressed on the cell surface. So, again trying to explain this codominancy in expression. So, these are the parents, let us say the male and paternal and the father and the mother, and you see that I put here, for this is just for explanation sake, you have HLA-A, HLA-B, HLA-C and HLA-DR. So, these are all the numbers that are given to various alleles that have been discovered and known to be present. So, these numbers signify a certain nucleotide sequence in the gene of that particular allele. W stands for workshop before it has been recognized as a definitive allele. So, if you look at these two sequences or these two alleles that are present in the father and the mother. Now, you have children, these children can have a combination, as you know from Mendelian inheritance. For example, here I have designated A 1 from the orange color is combined with the A 3. So, A 1 and A 3, or it can be A 2 and A 3, which is given over here. So, these are all tightly linked loci. So, many of these are actually coming together. So, you will see that A 2, C W, C 2, B 13 and D R 2 are inherited as a complex or because they are tightly linked. Occasionally, of course, many times of course, this sort of there could be a recombination within the MHC. Instead of D R 2, you can have D R 1, or if the parents are somewhat different, having a different allele, they can have different alleles coming in, and so you have a recombination within the MHC complex. But we will not go into that, just to show you, to drive home the concept that this sort of inheritance of these alleles, of the MHC alleles. Let us see what happens at the level of the cell surface. So, what you see over here is we have I have designated this green as the A 3 allele. So, if you take the parent over here, you find that the A 3 allele is expressed as, this is the class 1 molecule, this being the beta 2 M and this being the heavy chain. Both of them put together of course hold the peptide bound to its MHC binding groove, and this entire complex is recognized by the T cell receptor of the incoming or the recognizing T cell. This of course is the antigen presenting cell or the antigen bearing cell, which are whatever you can consider as. If it is class 2, it is a antigen presenting cell, and in the case of class 1, it is also known as antigen presenting cell, but many of these cells express these MHC molecules on the cell surface. So, what you see over here, which is now I have put it in a different color, it is trying to represent that this allele is different from the one in green. So, based upon what sort of allele is there, let us say for example, if you take this parent A 4, this designates A 4. So, you have both A 3 as well as A 4 class 1 molecules being expressed on the surface of this cell, and therefore, the peptide that is bound to this different MHC class 1 molecule or this different allele will also be different. And a T cell receptor from a non-self individual, or let us say you have a T cell receptor that recognizes A 3 as self, then the same T cell receptor will recognize A 4 as non-self. So, you see how the self and non-self recognition comes into play over here. So, in a certain individual, like for example, in this parent A 3 and A 4 being expressed on the surface of a single cell, the thymic differentiation has educated the T cells within that body to recognize A 3 and A 4 as self. So, therefore, when A 3 and A 4 present peptides, the T cell starts to react and recognize that peptide. In addition to recognizing this peptide associated MHC class 1, the T cell receptor also recognizes the alpha helices on the sides of this class 1, and therefore, is recognizing the MHC 1 also. In fact, that is the way it tells whether the MHC 1 is actually self or non-self. A non-self MHC molecule, which is now an allo MHC molecule, with its bound peptide is recognized as something as a different MHC molecule that is bound to the peptide by self T cell receptors. So, that is the concept of an allo recognition, in which case the allo recognition of course stimulates T cells in a much better way. So, therefore, these are the different kinds of alleles that are present within the children of these parents, and they are codominantly expressed. This is an example that I have shown you only for the HLA-A allele, but the same thing holds true for the other HLA alleles, whether it is BC or DP, DQ and DR. Now, this is just to show you how the peptide, as I told you earlier, is recognized by the variable regions of the T cell receptor, which recognizes this entire MHC class 1 molecule or the class 2 molecule if it is a class 2 mediated presentation. So, this is just to show you how the self is very important for recognition. So, if you have the same haplotype bearing self cells recognizing an infected viral antigen, then there is a killing. If there is no match within the MHC, then there is no killing. So, in addition to all these, you have of course other antigens playing a major role or minor roles in organ transplantation. So, as you see over here, this is the area where there is a lot of differences in the different alleles, and of course, within the floor of the MHC molecule you have differences in how different amino acids are replaced in different alleles. And these are of course just to show you how the peptide sits in that groove, and this peptide is actually contacted by the T cell receptor that comes from the top, and that is how the T cell receptor signaling occurs, because it is activated. So, this non-self MHC molecule is able to act as a self MHC molecule that is conjugated or bearing a pathogenic viral antigenic peptide. So, this is called as an allo response, to reiterate. So, in addition to all these kind of major histocompatibility reactions, you also have minor histocompatibility reactions. What are these minor histocompatibility reactions? In addition to these major histocompatibility molecules, you have various other kinds of what are called as non-classical MHC molecules. Non-classical MHC molecules, and it is practically impossible to match and try to match the tissue type for all these different kinds of antigenic major histocompatibility molecules. It was called as the major histocompatibility molecule because it is the major site or the major complex of antigens that determines skin graft or graft rejection reactions. So, these non-classical MHC molecules can also differ and have different kinds of alleles. In fact, they may not be as polymorphic as the classical MHC molecules, but they are polymorphic nevertheless. In addition to this major non-classical molecules, you have what you call as a minor histocompatibility antigen. So, these minor histocompatibility antigens are nothing but different kinds of antigens within the body. For example, we have several enzymes, they may have different kinds of isoenzymes or isozymes. So, many of these isozymes may have small differences in their sequences from person to person. So, these are different kinds of antigens that could get presented by the self T cell, presented by the self APCs or the antigen presenting cells, and you can have a reaction to that presented antigen. And therefore, you will see that many of these graft rejection reactions are usually evaluated majorly for 1 year, beyond which of course, if the graft is accepted very successfully, the studies that you will see in the literature goes on for about 8 years. But subsequently, you do see certain kinds of reactions setting in subsequent to 1 year, which is basically because all the different loci cannot be matched, and these are then controlled by different chemicals, which you will see as we go on, to immunosuppress the ongoing inflammatory or activated inflammatory responses. The better the medical means by which you can suppress immune responses, the better will be the prognosis of grafting procedures. So, what are all the different factors that affect skin grafts? So, you see some of the genetic factors, like for example, living donors, normally twins, okay, they are the best match that one can get. They share their haplotype, and therefore, there could be a perfect match. But how many of us can get twins as donors, or in the situation of accepting or taking skin grafts? So, cadaver donors are preferred. So, these cadaver donors should be HLA matched for obvious reasons as I have just now indicated to you. So, what is the immune status of what one does during this whole procedure? So, in addition to testing for the tissue type or trying to test what type of HLA molecules are there between these two organs or the cadaver and the recipient, you also have to type for the ABO blood groups. We will come to that in a little while. Also, one has to test for the presence of antibodies to the incoming HLA molecule or from the recipient organ. So, if the person already has for some reason antibodies that can recognize some glycosylated portion of the incoming HLA molecule from the graft, then you will have an immediate reaction against the graft. So, one has to pre-test the individuals or the recipients for anti-HLA antibodies. So, perhaps some of these patients might have been grafted earlier for some reason, and therefore, this becomes very much necessary. In addition, if they have undergone blood transfusion, people who undergo multiple blood transfusions actually develop antibodies to the small quantity or small numbers of T cells that have gone in with this blood transfusion. After all, blood, the RBCs are isolated within these medical facilities, but all these procedures cannot make them 100% pure. There will be a few T cells that will leak across and will be transfused to the person who is undergoing the transfusion. But of course, some of these T cells are not a major problem when you are undergoing blood transfusion or requiring blood, but they may cause problems because they will have elicited anti T cell immune responses over a period of time, and then when one has to get an organ, then you will have this T cell responses playing a major inflammatory role. So, the rejection of non-responsiveness must be monitored continuously in grafted individuals and controlled by using immunosuppressive drugs. What are these immunosuppressive drugs? These are ATG, will come to that, azathioprine will come to that in a little while, antibodies against T cells or total lymphoid irradiation. That means, you irradiate the individual in order to inactivate the T cells from proliferating. So, in somewhat you cut down the extent of the immune response because you have stopped the proliferation of T cells, but of course, if the cytokines can cause some damage. But this total lymphoid irradiation is actually now not being preferred as much as they were in the earlier days. So, matching for HLA-B, HLA-DR play a very great role in graft survival. So, relative to HLA-A and C, HLA-B and DR need to be considered much more. Immunosuppressive regimes which involve drugs like cyclosporin A along with azathioprine, which is A in this ATG, steroids and anti-thymocyte globulin must accompany these procedures. So, these kinds of interventions, they are to be used during acute episodes meaning that as soon as one has the graft, you see the rejection reactions because of some problem. So, there has to be hypersensitivity testing for allergic reactions to the incoming graft, because that is one part of inflammation. Anti-CD3 is useful only in these acute episodes. Now, CD3 is the anti-lymphocyte antibody, because CD3 is the T cell receptor. T cell receptor has the CD3. So, if you have antibodies to the T cell or the anti-CD3, it cuts down on the function of these T cells. By using this anti-CD3, you can also deplete these T cells because of complement mediated reactions, but basically it inhibits the function of T cells by using these anti-CD3, and this is possible. Anti-CD3 is known or documented only to be very useful when there are acute episodes rather than a chronic rejection episode that is occurring over a long period of time. Anti-IL-2 receptor this IL-2 receptor is very important for T cell proliferation. So, if you were to use antibodies to this IL-2 receptor, you can cut down on the proliferation of T cells and therefore, less T cell help to the T cells that are reacting against the graft. Some of the addition molecules like LFA1. So, anti lymphocyte function associated one molecule, you have antibodies to that, you can actually inhibit the migration of many of these cells and therefore, they do not go into the proper location. Now, Campath1 is an anti-lymphocyte CD52 antibodies which have been used with a fair degree of success in transplantation reactions. So, when you look at other immunological factors, of course, total lymphoid irradiation using X irradiation was used for Hodgkin disease. It is more useful in the case of kidney allografting, but is now less widely used. Anti-class 1 antibodies causes hyperacute graft rejection HGR. This is in fact a term that is used in transplantation studies. So, if there is anti-class 1 antibodies already present within the individual before the grafting occurred, then you have a very rapid graft rejection reactions. Anti-class 2 is less severe in causing hyperacute graft rejections. So, anti-lymphocyte auto antibodies of course can be present, but they do cause problems, but not as much problems as anti-class 1 antibodies. So, of course, you have to screen for anti-monocytes and anti-endothelial antibodies which are poor prognostic indicators of good skin graft acceptance. As I told you earlier, cross matching is a must. Cross matching for antibodies is a must, and it must be countered if there is presence of these antibodies, it must be countered by intravenous immunoglobulin or what you call as IVIG treatment. Now, in renal disease, one can make out if there is a hyperacute graft rejection, if there is anemia, and of course, it is treated by recombinant erythropoietin and blood transfusion, and the blood transfusion has shown to be beneficial to decrease graft rejections for a variety of reasons. Now, if you go on further, as I told you earlier, minor histocompatibility antigens play a very great role. So, graft rejection is primarily a T cell mediated reaction. Now, advances in immunosuppressive therapy actually holds the key. Xenotransplants using pigs may be an alternate source of organs. So, transgenic pigs that express human decay accelerating factor or pigs lacking alpha gal are being developed in order to see how these different kinds of organs can be used in human transplantation, because in humans antibodies prevail against this alpha gal moieties, and therefore, they are preventing these sort of organs being used from the pigs. Now, the grafts are evaluated as I told you earlier for 1 year, but eventually most grafts are chronically rejected. Chronically means slowly being rejected over a long period of time by about 8 years. Now, many of the reasons for graft rejection are arteriosclerosis, meaning blockage of blood vessels, ischemia, reperfusion injury that occurs when you are actually suturing the graft inside, because you are cutting the blood vessel and then suturing them. So, ischemia means blockage of blood supply, and then you open the blood supply again, and that is reperfusion, which means now the blood starts to flow. Ischemia itself and reperfusion itself are known to have a lot of deleterious effects within the body. Cyclosporin is an immunosuppressive drug, but it has its own toxicity. And of course, you have cell mediated reactions, macrophage infiltrations into the graft, which causes all these different kinds of graft rejection reactions. And of course, there are different kinds of chemokines, chemokine ligands, all of them participating in these inflammatory reactions. For want of time, I would like to skip some of this information, but they will be available for you in the slide. Now, some of the another interesting feature or important feature to be considered is that leukemias and lymphomas, they are blood cancers, they require bone marrow transplantation. Okay. This is how they actually try to address or combat people who have leukemias and lymphomas, because they are highly proliferative lymphoid cells. Of course, there are different kinds of leukemias. This bone marrow transplantation is actually done to provide hematopoietic stem cells. So, these hematopoietic stem cells proliferate within after giving the bone marrow, they proliferate and then repopulate the person's immune system by providing them with new lymphoid cells. Of course, these hematopoietic stem cells have got to be normal. In fact, nowadays they are trying to see how stem cell therapy can replace many of these kinds of medical treatments, because the stem cell itself does not express MHC molecules. So, you have stem cells then differentiating to form different kinds of blood cells. As these blood cells are differentiating, tolerance sets in, and that could perhaps mean that there could be a longer time for a particular graft to survive. T cells in the donor tissue can react to the recipient, which we are studying in this lecture, since the patient will be in a immunocompromised state due to irradiation and chemotherapy. So, during this whole procedure, in order to prevent the rejection of the graft, the patient has been treated with immunosuppressive drugs, be it steroids or cyclosporins. So, it gives a chance for this bone marrow. The bone marrow has lymphoid cells and therefore, may have T cells. So, you have T cells reacting against the recipient. So, the donor T cells now starts to recognize the host and starts to react against the host, because the host is immunocompromised. The host can also be immunocompromised if you irradiate the host, because the host, the patient has got a tumor like leukemia. So, this is called as a graft versus host disease, GVHD. So, here donor T cells proliferate and they form cytotoxic T killer cells due to allostimulation, because these donor T cells recognize the host T cells as foreign, but the host T cells cannot react to the donor T cells, because they are immunocompromised due to irradiation or immunosuppression. The cells may kill these leukemic cells. This is called as the GVL graft versus lymphoid effect, which is in the medical field they understand what is this GVL effect, which is basically that the T cells coming from the donor graft, they kill the leukemic cells, because they can recognize the leukemic cells. For example, NK cells, natural killer cells, can recognize tumor cells, especially leukemic cells, which are decreased MHC antigen. In fact, that is one of the ways by which NK cells recognize and kill their target cells. So, they can kill. So, there is a little beneficial effect of these donor T cells, which have been activated from the donor grafted tissue. GVHD is mainly due to the recipient's dendritic cells. So, the host dendritic cells take up some of the antigens from the grafted tissue and present it to the donor T cells. Hence, the recipient's dendritic cells need to be depleted. So, these are all the strategies that one follows during these kind of transplantation, bone marrow transplantation procedures. Expansion of Treg cells, as I told you earlier, regulatory T cells are immunosuppressive T cells, they suppress cytokine release. So, many of these T cells, Treg cells, if they are expanded, then you have a good prognosis. So, they take out these they expand them ex vivo, that is now being tried to inhibit graft versus host disease. Now, these Treg cells can be purified based upon the expression on the cell surface, which are the CD25. So, they express CD25 on the cell surface. So, CD4, CD25 positive cells can be purified by a variety of techniques, grown in vitro or using tissue culture, and then reinfused into the patients. So, what are these immunosuppressive drugs? So, corticosteroids, that is known to be all of us know that they are used for immunosuppression. So, for example, prednisolone. So, you see IL-1, TNF or tumor necrosis factor, IL-3, IL-4, IL-5, CXCL8, they are all decreases when corticosteroids are administered. Therefore, this decrease in these molecules actually lead to reduced inflammation. Another molecule called as inos, nitric oxide synthase, it also decreases when corticosteroids are injected. This leads to decreased NO production and therefore decreased inflammation. In addition to PLA2, phospholipase A2, cyclooxygenases, they all decrease when corticosteroids are injected, and it leads to reduced prostaglandins and leukotrienes, and therefore decreasing the ongoing inflammatory response against the graft. Endonucleases, which cleave up the DNA, they increase, indicating apoptosis. So, corticosteroids actually increase apoptosis of many cells. They decrease adhesion molecules, therefore, lymphocyte trafficking also decreases, reduces cell migration. So, macrophages coming to the vicinity of the inflammatory site will be decreased. Some of the negative aspects of course, are fluid retention, weight gain, diabetes, bone loss and skin problems. Azathioprine is a chemical, it is metabolized to 6-thioinosinic acid. It blocks AMP and GMP synthesis. In fact, that is how it blocks proliferation of treated cells and hence DNA synthesis. It blocks CD28 co-stimulation of T cell immune responses. T cell, in addition to the primary response, primary stimulation, it requires a co-stimulatory signal coming from CD28, and it accelerates apoptosis by blocking the small GTPase called as RAC1. The bad effects are anemia, leukopenia, damage to intestinal lining, hair loss, decreased immune function and mutations, which is a very bad aspect of azathioprine treatment. Similarly, mycophenolates are used to inhibit IMP dehydrogenase and GMP, which ultimately leads to blockage of proliferation. Cyclophosphamide is another drug which is metabolized to phosphoramide mustard, which alkylates DNA. The bad effects are hemorrhage and neoplasia of the bladder. Cyclosporin, which is the most important or most popular drug that is used for immunosuppression, it is a cyclic decapeptide. It is actually got from a soil fungus called as Tolypocladium inflatum. It is named as tacrolimus, FK506 is the other name. It is a macrolide from the filamentous bacterium Streptomyces stucabensis. It is a many membered lactone ring to which is attached one or more deoxy sugars. The other drug is rapamycin, which is called as sirolimus. It is also a macrolide from another Streptomyces organism. All these act by binding to what are called as immunophilins, and they alter signaling within cells, transmembrane signaling events. Cyclosporin and FK506 block T cell division by inhibiting calcineurin, calcium activated phosphatase. Calcineurin is a calcium activated phosphatase. IL-2 synthesis is blocked and therefore, proliferation of T cells are blocked. The toxicity of cyclosporin is actually shown on kidneys and other organs, and one of the most problems with using cyclosporin is of course, it is very costly. There is another the other drug rapamycin inhibits mTOR. mTOR is a serine threonine kinase which is involved in PI3 kinase AKT pathway, which is operative for T cell division and apoptosis. Of course, monoclonal antibodies have also been used in transplantation, like for example, Campath H, as I told you earlier. It has been used for solid organ and bone transplantation grafts and for the elimination of mature T cells, because it binds to T cells from the donor bone marrow. So, a GVHT can be inhibited. OKT3 antibody against CD3 causes immunosuppression by inhibiting the T cell receptor. It has been used for solid grafts and causes unwanted cytokine release because of Fc dependent activation of Fc receptor bearing cells. So, they have used a mutant which is an mutant where alanine has been replaced in the normal sequence of the CD3. This is used to prevent this, but use of this antibody is less frequent. In animals, non depletion of anti CD4 treatment is used to induce antigen specific T regulatory cells. So, non depleting meaning that this anti CD4 antibody has the property of not causing toxicity or not being cytotoxic to the CD4 cells. Apparently, this sort of antibody enhances or induces Treg cells. Similarly, CTLA4 participates in the co-stimulatory signal of T cells. This CTLA4 has been used to block CD28B7 co-stimulatory interactions, and that is how it causes blockage of T cell stimulation. Anti-CD40 ligand is another very important strategy that has been used in primate kidney transplants. Of course, all this needs to have the HLA being typed, and this HLA typing procedure uses antibodies to see whether there are allele specific antibodies. So, you have specific anti-sera that recognizes specific specificities, and using these plates you can look for staining of these cells by using different kinds of dyes, which has been enumerated in the MHC class. And these various kinds of HLA typing procedures, this is one of looking at the microcytotoxicity. Then you have what is called as RFLP or restriction fragment length polymorphism, using different restriction enzymes to cut up the DNA of different individuals and then trying to probe it with a particular probe that will bind to a particular HLA allele. So, to summarize all these studies, you find that all these graft rejection reactions, rather than trying to explain the different procedures that are involved in graft rejection reactions and how they are evaluated, because that is more clinical in nature, I have tried to focus on some of the basic principles that are involved in explaining why an allotransplantation reaction ensues if the graft has not been typed or matched. So, basically then, if you look at graft transplantation reaction, one has to type the tissue, and this typing of course is done to match the HLA types on the incoming or the donor the donor receiving the graft. So, both of them have to be HLA typed, and of course, nowadays since there is more difficulty in getting this matched tissue grafts available, one resorts more and more to try and see how you can improve the techniques that will immunosuppress the ongoing immune responses. So, thank you very much, and we will go on to other lectures in the future.